dear colleagues, very good morning. My name is Rambabu Korpati. Today I am going to present my research titled Optimizing Artemia Life Stage Utilization for Improved Performance of Lithopenes lanami post-larvae. Well, coming to introduction, L. lanami is commonly called as Pacific white shrimp. It is one of the topmost aquaculture species in the world. During early larval stages, it is commonly fed with Artemia, commonly called as brine shrimp. Artemia is having a lot of advantages like high protein content, better nutritional value, easy to store, ready to hatch, etc. compared to other live fish such as Moina and Daphne. During unfavorable conditions, the adult female lays cysts. Once we provide favorable conditions like optimal temperature, salinity, continuous aeration and light, it gets hatched into free-swimming larvae called Install-1 Artemia. Farmers in Mexican regions and Caribbean regions and Ecuador regions, around these places, they were feeding decapsulated Artemia for the shrimp during early stages. They are removing the outer layer of the cyst and feeding them to the post-larvae. But some farmers in Asian regions, they were feeding Install-1 live Artemia. They believe that this live Artemia has some active bioenzymes which helps in maximizing the growth and other factors and have better disease resistance in L. lanami. There is the global shrimp industry requires a valuable data and authentic data in order to make sure which form of Artemia is better in terms of providing maximum survival, growth, disease resistance, and healthy gut microbiome for L. lanami. In order to fulfill this research here, we came with an experiment where our objective is to identify which type of Artemia, whether it might be live, decapsulated, or maybe combinations, which is good for maximizing the growth, survival, stress response, and gut microbiome diversity of L. lanami post-larvae. We came up with an experimental design where we have followed complete randomized design where I have 4 different diets with 6 different replicates for each diet. We have conducted power analysis in order to make sure we have enough replicates. Our diet 1 is 100% live Artemia and diet 2 is 100% decapsulated Artemia, where diet 3 and 4 are combination diets. We have measured the water quality both before and after feeding. And for gut microbiome analysis, we have done 16S RNA sequencing. For measuring the stress physiology, we have done heat shock protein gene expression analysis. If we see the graph 1 and graph 2, they represent the water quality of pH before and after feeding, where we can see they lies between 7.9 to 8.5. The pH lies between 7.9 to 8.5, which is optimal level for L. lanami during the early stages. And graphs 3 and 4 shows total ammonia, which has both ionized ammonia and unionized ammonia. We have measured the ammonia by using API test water kit. Even though there are some fluctuations in the ammonia, but they are within the optimal limits. This suggests that the diets did not influence anything on the water quality. All the changes happened in the L. lanami is only due to the diets, not due to external factors like water quality. If you see table 1, 2, 3, shows the consolate composition, fatty acid composition, and amino acid composition of Install-1 Artemia. As mentioned earlier, Artemia is having a lot of advantages. It is having high protein content, essential fatty acid concentrations are higher like arachidonic acid, icosapentaenoic acid, glucoshexaenoic acid, and it is also having higher levels of essential amino acids like cysteine, lysine, leucine at higher levels compared to other live feeds like Moina and Daphne. So that's why most of the farmers, like 90 to 95% of the hatchery farmers, use Install-1 Artemia or other forms of Artemia as a food source during early development stages for banana. Well, coming to methodology, for gut microbiome, we have done 16S RNA sequencing, where we have collected the guts from the shrimp, extracted the DNA, and then we have performed 16S RNA sequencing amplified at V3, V4 hypervariable regions. We have done bioinformatics using CHAOS-1, and for statistical analysis, we have done observed species indices. And then we have done taxonomic classification of bacteria using LEFC. While coming to gene expression analysis, we have collected the hepatopancreas region of the shrimp. We have extracted RNA by using Iazin kit, and from RNA is converted into cDNA by using the thermocycler through reverse transcription process. And we have loaded respective amounts of cDNA, forward and reverse primers of heat shock proteins, and we have done amplification in the PCR, where we obtain delta cycle threshold values. With these values, we have converted into graphical results. Measuring the tiny shrimp is very difficult. We have to take them out from their aquatic habitat. Traditionally, they have done that. We have to take them from aquatic habitat, handle them with our bare hands. It is very stressful process for them, and also there is no scale to measure that tiny shrimp. So in order to overcome this, we came up with an AI-based image processing technology where we need to do only one thing: just capture an image. Once the image is captured, the image will be getting uploaded into the cloud network, where image gets processed by forming horizontal and vertical imaginary lines by using CNN and YOLO technologies. Once the imaginary lines formed within the cloud, it calculates the number of segments the shrimp occupies within those imaginary lines and gives the lengths and weights as shown in figure 6. If we see here, we can see the number of shrimp like 244 was the average length, was the average weight. Based on this, we have analyzed our growth data. While coming to stress physiology, we have done gene expression analysis using the primers listed in table 2. Heat shock protein 70 and 90 usually acts as a biomarkers for stress. Heat shock protein 60 helps in protein unfolding at mitochondrial region. And as we know, if there is higher heat shock protein expression, that means that particular shrimp showed higher heat stress resistance. But in contrast, we can see here diet 1, which is 100% live, showed least heat shock protein expression. That means the diet is having less stress resistance. These particular treatments show least stress resistance. Whereas in contrast, the diet 3, which is having 75% live and 25% installment, showed maximum heat stress resistance. Whereas we observe carefully, diet 2, 3, and 4 is having decapsulated artemia in common. Decapsulated artemia has beta glucons present in the outer layer of the cyst. This might be responsible for these elevated heat shock protein expression levels in these treatments, and because of that, there is no decapsulated artemia in treatment 1, so that's why the heat shock protein levels are very less, and these particular treatment perform very low heat shock protein stress resistance. While coming to our alpha diversity measures, diet 1, which is 100% live, showed maximum gut microbiome diversity compared to other diets, whereas diet 2 also showed some better diversity compared to the mixed diets, combination diets. But these diets is not completely providing. They are mostly opportunistic bacteria which are very harmful for the gut, which is proved in figure 8 heat map, where we can see the diet 2 entire column is mostly stable except these particular regions where we have mostly opportunistic bacteria. This is also proved in the cladogram shown in figure 9, where diet 2 is represented in green color, where we can see all of harmful families like Aeromonidaceae or Aeromonidales, Enterobacteriaceae or Enterobacterials. All these are harmful bacteria, they are not useful bacteria. Our left C analysis also showed that diet 2, which is 100% decapsulated artemia, is having higher LDA score. That means they are having high gut microbiome diversity. That doesn't mean they are having high probiotic bacteria. They are mostly opportunistic bacteria. If you see like Gamma bacteria, Aeromonidales, all these are harmful bacteria. Our rare fraction analysis has showed that we have done complete RNA sequencing coverage, and all these plateau lines suggest that we have done enough RNA sequencing coverage. While coming to the growth data, as we measure the growth using the artificial intelligence image processing technology, we can see diet 1, which is 100% live artemia, performed very well in terms of growth and survival, whereas diet 2, which is 100% decapsulated artemia, performed good in terms of growth and survival. While coming to conclusion, our growth data suggests that 100% live artemia showed better growth and survival compared to 100% decapsulated artemia, whereas heat shock protein analysis suggests that inclusion of decapsulated artemia not only increases the stress resistance but also decreases the cost effectiveness for the farmers. And our gut microbiome diversity analysis shows that diets having 100% decapsulated artemia are having mostly opportunistic bacteria, whereas diets containing live artemia is having beneficial bacteria like Lactobacilliaceae family, Vibrionaceae family. So I suggest for the farmers in Mexican and Caribbean regions to not to use 100% decapsulated artemia, at least include 75% of live artemia so that they can improve higher growth, survival, and gut microbiome diversity. And I also suggest for the farmers in Asian regions to not to use complete 100% live instar artemia, at least include 25% of decapsulated artemia, which not only increases the stress resistance but also decreases the cost for their production. Here are my references used for the study. I need to thank my professor Dr. Etish Ramina, Dr. Grace Ramina, Dr. Tomeka White, Dr. Adam Fuller for being my members of thesis committee and supporting throughout this project. I need to thank USG and NIFA for funding this project. I need to thank Great Salt Lake Artemia, Infinicy, and Keaton Industries for supporting throughout this project. I'm happy to to take questions.
Would you go back to that ammonia graph that you showed? Oh, it's on page two. How do you explain though, you said there were but do you have any explanation for those speeds that you see?
Here, the graphs 3 and 4 are total ammonia. That doesn't mean like they are unionized ammonia. They are having both ionized ammonia and unionized ammonia. Unionized ammonia is not harmful. Usually, the best water kit which the API provide, which is a standard kit used by all over the aquaculture industries, we're having limits over the chart which they provided, they are within the range. And I haven't seen, if we see the growth data, we have stopped 250 post-larvae, and we have seen maximum growth of 244 at the end of the experiment in one replicate. That means they are already within the limits.
That's what I'm looking for.
Yeah, we have used convolutional neural networks, and you only look once models by using Python code.
Okay.
Yeah.
So did you validate it with the AI models that we actually trained after we were trying to quantify?
We have tested that AI using in collaborated with Arkansas Tech University. They also got the same results.
Okay.